Welcome to lecture number 14 of the Genome 361 class. Today we'll be talking a little bit about cancer genetics. So I'm going to depart from my usual tradition of going through the key points of today's lecture. You can come back to them at the end of this lecture to make sure you understand all the key points. I'm just going to skip ahead to the individual who's shown in the photograph at the bottom of the slide. So this is actually Harold Varmus, who won the Nobel Prize in 1989 for his discovery of the cellular origin of a type of cancer gene known as an oncogene. We'll talk more about oncogenes later in the lecture. But the main thing I wanted to focus on here in this slide is the statement that's attributed to him, where he says, "Cancer is a collection of many diseases with common principles, and each disease will have to be understood and more effectively controlled on its own terms." So I think that this statement is best illustrated by the 5-year survival rates and the change in those 5-year survival rates that has occurred in various types of cancer over the past, say, 25 or 30 years. And that's actually shown on the next slide. So this slide shows the 5-year cancer survival rates in the US for a variety of different kinds of cancer. And basically, for most of these types of cancer, if you look at the left end of each of these arrows, that's actually what the 5-year survival rates were in around the mid-1970s. And typically, for most of these arrows on the right is what's shown is the 5-year survival rates of these cancers around 2007 to 2013. Now, I'm not going to go through all these. I just want to make a couple of general points. So first of all, I've highlighted a few of these here to show that, in fact, the 5-year survival rates for a number of these cancers have gone up dramatically from the mid-1970s up to close to the current time. So, for example, prostate cancer at the top. The 5-year survival rate was at one time about 67%, 68%. Now it's almost 100%. And that's true, at least the 5-year survival rates have gone up to a similar degree for a number of these cancers on this that I've highlighted here. But despite the progress that's been made on these cancers, what you can also see is that, in fact, for some of these cancers, for example, uterine cancer, the 5-year survival rates actually decreased a little bit. Or for pancreatic cancer at the very bottom of this list here, the 5-year survival rates have actually increased, but pancreatic cancer still remains a very lethal cancer, only a 8.2% 5-year survival rate for people with that cancer as of around 2007 to 2013. So what explains all this variation? Well, the part of the explanation for the changes in the 5-year survival rates is that there's been better early detection for some of these cancers, and there's actually been better treatments for many of these cancers. But that actually isn't true across the board. In some cases, the detection methods have not increased or improved over that period of time, and the treatments that work for some cancers just don't work for other cancers. So again, going back to the statement that Harold Varmus made, where he says cancer is a collection of many diseases with common principles. Importantly, on the previous slide, the point that's best illustrated on the previous slide is that each disease will have to be understood and more effectively controlled on its own terms. So basically, when somebody says, "I hope they'll develop a cure to cancer," that will actually never happen. What people are really saying is, "I hope that they'll develop a bunch of cures to the various types of cancers that are out there," because each cancer is a bit different. The genes involved in the cancers are different from one cancer to the next, and so they all really have to be treated separately and dealt with as a separate matter. However, the first part of Harold Varmus' statement is also true. Cancer is a collection of many diseases with common principles, and it's actually the common principles that we'll be talking about in today's lecture. So just a few statistics about cancer in the US. So there's about 1.25 million new cases of cancer each year. It accounts for about 550,000 deaths each year. That's about 25% of all deaths. The lifetime chance of developing cancer is nearly 50% for men. It's about 38% for women. And the lifetime chance of dying from cancer is about 24% for men and about 20% for women. So what is cancer? In simple terms, it's just unregulated cell growth. And actually, the process of forming a cancer cell is a multi-step process. So it's basically a progressive loss of cellular homeostasis or balance. And it begins when a single cell, has an altered growth control, and that cell starts to divide in an uncontrolled fashion, and it forms a hyperplasia, which is basically a pile of cells. And then this eventually becomes a dysplasia, which is basically means that the tissue or the cell has an altered morphology. And then that progresses to an in situ cancer, which is basically the cells have acquired the cancerous state, but they're still trapped inside the tissue of origin. They haven't moved. And ultimately, the final stage is metastases, where the cancer cells move from one tissue or organ to another tissue or organ. And the progression through all these stages is actually caused by accumulated mutations. So it starts with a single mutation, and then the cell that started this whole process acquires additional mutation and then additional mutation, and this just triggers the process all the way through to the cancer state. We'll talk more about that later in the lecture. So what exactly is the evidence that mutations cause cancer? So the first demonstration that cancer is actually a genetic disease was actually through experiments that were performed in the early 1980s. And the experiment that was done was people asked whether DNA from cancer cells could convert, if you transfer that DNA to a normal cell, could it convert a normal cell to the cancerous stage? So basically you take tumor cells, recover the DNA from those cells, transplant that DNA into non-tumor cells, and then ask, do the cells that have received DNA from the tumor cells, do they become tumor cells themselves? So this is the experiment that people did in the 1980s to kind of work out that cancer is caused by mutations. And we'll describe the experiment that was done first on the next slide. So basically what people did is they took mouse cells that were growing in culture, and typically you can grow a variety of different cells from a variety of different organs in culture. And typically what happens when you do that, the cells will divide until they sort of contact one another, and then they stop dividing. So that's known as contact inhibition. So the cells will just grow until they reach their neighbors, and then they stop growing. They can receive signals that tell them, okay, there's enough cells here, stop dividing. And what they did is they took these cells and they introduced into the cells DNA from human bladder cancer. So they just took fragmented up DNA from a human bladder cancer. So there were just large fragments of DNA from the human bladder cancer cells. And they introduced them into these mouse cells, these normal mouse cells. And what they discovered when they did that is that these cell foci would arise periodically in the culture dish. And what a cell foci basically is a cell, it's a clump of cells that have lost contact inhibition. So the cells, even though they've contacted their neighbors, they continue to grow and divide. And then what people found was if you took these cell foci and you inject them into mice, the mice developed cancerous tumors. So the conclusion from this experiment was that there was some sort of genetic element that transformed these normal cells into a cancer cell because they had some sort of mutation or gene altering mutation that was that caused the cancer. So if you were doing this experiment, how would you identify the gene responsible for transformation? So if I had a live audience here, I would basically ask you guys to kind of come up with ideas, but I'm just going to walk you through the different steps that you might go through if you were going to try and identify the gene responsible. So basically what people did is they fragmented the DNA from a human bladder cancer cell, and they made a library with it. So they introduced it into plasmids essentially. And then they basically introduced that library of DNA into these normal cells, these normal mouse cells. And then they isolated DNA from the cell foci. These are the cells that have lost contact inhibition. And then they compared the sequence of the DNA in the cell foci to the corresponding sequences from normal cells to look for gene-altering mutations. And what they found when they did this kind of work was they found there was just really only one difference, and the difference was a single base change, a G to a T, that results in a glycine to a valine change in one single gene called the CRAS gene. So how does this mutation in the CRAS oncogene cause cancer? So cancer really just comes down to a loss of cell cycle control. Now, I know you've talked about the cell cycle in the first half of the course, so this is hopefully a review for you. But basically, the cell cycle can be divided up into 4 different stages. Basically, the first stage is just the time when the cell is growing normally and not trying to divide. That's called G1 or gap 1. The next part of the cell cycle is S phase. That's when DNA synthesis takes place to prepare the cell for cell division. Then G2 is the gap between the end of DNA synthesis to prepare the cell for mitosis and the beginning of mitosis itself. and the fourth stage of the cell cycle is mitosis itself. So it turns out there's actually positive regulators and negative regulators that act at two key points in the cell cycle. They act at the point where the cell is actually trying to decide to initiate DNA synthesis, so it's making a decision whether it should start to initiate the process that leads to cell division. And the other place where these positive and regulators and negative regulators act is at that point between G2 and M, when the DNA synthesis is complete, but the cell is deciding whether to enter mitosis. So it turns out that the CRAS oncogene is a positive regulator. Its job is normally to promote entry into the cell cycle at both of these 2 points, between G1 and S and between G2 and M. The normal non-mutated versions of an oncogene are called proto-oncogenes. So we all have proto-oncogenes, and they only become cancerous if they undergo some sort of mutation. We'll talk about the 3 types of mutations that could occur to an oncogene or a proto-oncogene to convert it into an oncogene on the next slide. So this slide shows the 3 ways in which a proto-oncogene can be converted into an oncogene. So what I'm showing you here is the proto-oncogene. Let's just imagine the red part corresponds to just the coding sequence of the oncogene, the protein coding sequence, I should say. And one way a proto-oncogene can be converted into an oncogene is through some sort of coding sequence mutation. So this can actually lead to a hyperactive protein, for example. And a good example of that is the CRAS oncogene, where a glycine that's normally present at a particular position in the protein coding sequence gets converted into a valine, and that single amino acid change converts the protein from its normal form to a hyperactive protein. So now it highly activates entry into the cell cycle. Another way this can happen is through gene amplification, where instead of having just 1 copy of the proto-oncogene, now you have 3 copies of the proto-oncogene. So it's basically just a normal protein that's being made, but the protein's being made at much higher abundance. So you end up having way more activity from the proto-oncogene than normal. And then the third way this can happen is through a chromosome rearrangement. And one way this can happen is where you can have a fusion of a strong promoter from some other gene to the proto-oncogene. So now you're overproducing the protein. You're overproducing the messenger RNA, and therefore the protein is also being overproduced. So it's just the normal sequence of the proto-oncogene, but you're making too much of it. And sometimes the fusion of the strong promoter doesn't involve just the fusion of the promoter itself, but it can involve the fusion of a strong promoter and part of the coding sequence of the gene that's normally coupled to that strong promoter to the oncogene. So fusion of a highly expressed gene that leads to the overproduction of a fusion protein, a protein that consists of the oncogene and some other protein. So these are the 3 ways in which a proto-oncogene can be converted into an oncogene. All 3 of these ways basically result in the same thing, a hyperactivation of the normal protein activity that's associated with the proto-oncogene. In the last lecture, I talked to you a little bit about the work that was done by Mary Claire King to map the BRCA1 breast cancer gene, and I told you I'd come back to this gene a little bit in the cancer lecture, so I'm coming back to that now to talk about this a little bit. I also told you the BRCA1 gene was identified, it was ultimately cloned and sequenced, and this is some of the evidence we talked about in the last lecture, to say this was actually probably the gene involved in breast cancer. So what does the BRCA1 gene do? So now I'm going to return back to the cell cycle, and we'll talk about what it does on here. So I mentioned in an earlier slide that the RAS oncogene acts as a positive regulator. It turns out the BRCA1 gene also acts in the cell cycle, but it's a negative regulator of the cell cycle. And negative regulators of the cell cycle are also called tumor suppressors. And many of the tumor suppressors act at checkpoints, what are called checkpoints of the cell cycle. So the checkpoints in the cell cycle I've defined a little bit better in the next slide. So cell cycle checkpoints are cellular surveillance systems that monitor the integrity of the genome and of the cellular structures. And it monitors these things because it wants to make sure that we don't enter mitosis, that the cell is not primed to really be ready to enter mitosis. So they enforce the correct order of execution of cellular events. They prevent cell cycle progression until genome quality control is assured. And the goal here is really to make sure that the genome, that the integrity of the genome is intact, and the cell is ready to go through the entire process of the cell cycle and to undergo cell division. So examples of the kinds of things that cell cycle checkpoints look at are whether the chromosomes are attached to the spindle. So if the chromosomes haven't fully all attached to the spindle, then cell cycle checkpoint control regulators block the onset of anaphase. They also look to see if the DNA is damaged, and if the DNA seems to be damaged, cell cycle checkpoint regulators halt the cell cycle to allow repair to take place. And if there's irreparable DNA damage, cell cycle checkpoint regulators will trigger cell death. And in fact, you can actually see some of the effects of loss of these cell cycle regulators in the karyotype of cancer cells. Because many cancers have mutations in genes that act at checkpoints, you can oftentimes see the effects of these mutations on the karyotype of the cancer cell. So what I'm showing you here in this slide is that this is a normal cell karyotype, and the color coding is because, in this case, a series of probes that recognize different chromosomes that are color coded for the various chromosomes they recognize were used to hybridize to the DNA from this particular cell. So all the different chromosomes can be distinguished from one another based on their color. So this is a normal cell. This is what a karyotype of normal cell looks like. So it's an XX cell, so you see 2 copies of each chromosome. But if you do this kind of analysis in a cancer cell, the karyotype oftentimes looks very different. So for example, here's another XX cell, but this is a cancer cell, and you can immediately see that the karyotype is altered in a number of ways. So for many chromosomes, you don't see the normal number of chromosomes. You see more than 2. Sometimes you see less than 2. Not only that, but you see a number of other alterations. Some of the chromosomes are shorter, indicating they have deletions. Some of them have translocations of other chromosomes onto the chromosome. And then if you look more closely, you'll see that a number of them have rearrangements. But basically what you'll see in cancer cells, because oftentimes cancer cells have mutations in genes that act at checkpoints, you'll see the karyotype is greatly altered simply because the cancer cell is now going through the cell cycle before all the proper checks have taken place to make sure the chromosomes are ready for cell division and the cell itself is ready for cell division. Okay, just a little bit about proto-oncogenes and tumor suppressor genes. So these are, if I haven't said this already, these are the 2 major types of cancer genes, proto-oncogenes and tumor suppressors. So in a diploid cell, the proto-oncogenes promote cell growth and proliferation, and tumor suppressor genes oppose cell growth and proliferation. So if you have a mutation in a proto-oncogene, basically what that's going to do is it's going to upregulate cell growth and proliferation. And a mutation of this sort is called an oncogene. And then also if you have loss of function mutations in the tumor suppressor genes, that'll actually lead to the increased cell growth and proliferation of the cell that bears these mutations. All right, so here's a question for you. And again, you can pause the video if you want to sort of take a little bit of time to think about this. But let's just imagine you have a cancer-promoting form that's of an oncogene. What's the expected behavior of an allele of an oncogene? Would you expect it to be dominant or recessive to the wild type? So again, you can pause the video if you want and think about it. But the proper answer is that it's dominant. And the reason it's dominant is because it doesn't really matter what the other copy of the proto-oncogene is doing. If one of the proto-oncogenes has undergone a mutation to convert it to an oncogene, such that, for example, it's overproduced or it's more active, it doesn't really matter what the wild-type version of the gene is doing. The activated form is still going to promote excessive cell growth and proliferation. How about mutated tumor suppressors? So in this case, you would expect these to be recessive. And the reason you'd expect these to be recessive is because if you mutate one of the 2 tumor suppressor genes, you still have the other one functioning and preventing cell growth and proliferation. But it's only after you've lost both of them that you've really lost that control, and the cell growth and proliferation starts to, amplify and lead ultimately to the cancerous state. So people have actually tested whether tumor suppressors and oncogenes exhibit a dominant or recessive behavior by doing what are called cell fusion experiments. And the way this works is basically, let's imagine you have a tumor cell, some sort of cancer cell, and if you inject that cancer cell into mice, what you'd find is that cancer cell will lead to the formation of tumors in the mice that were injected by it. Whereas if you take a normal cell, a non-cancerous cell, and you injected that into mice, you would not expect to see tumors. It turns out there are actually chemical agents that will allow the plasma membrane of cells to fuse with one another. And when you treat cells, like if you take a cancer cell and a non-cancer cell, you treat them with these chemicals, this could lead to cell fusion. So now instead of having two cells, the two cells have fused together and you have one. And this allows people to test whether the cancerous cell has a mutation that's either dominant or recessive. And the way you do this is then you inject the cell into mice and you ask whether tumors form. Okay, so if you think about this, what's the prediction? If the cancer is due to an oncogene, would you expect tumors to form or not? And the answer is tumors would in fact develop if the cancer was due to an oncogene. Because again, when you fuse the cells, you basically have some copies from the cancerous cell, at least one copy of the oncogene present. So it doesn't really matter what the normal versions of those genes are doing. As long as you have one copy of an oncogene, a tumor is going to develop because you're going to have excessive activity from that one oncogene. Now, if you do the same experiment with a tumor suppressor, what's the expectation in this case? So again, the expectation is you'd see no tumors because even if you have two mutated copies of the tumor suppressor gene in the cancer cell, once you do the cell fusion experiment, you're going to have two normal copies of the tumor suppressor from the non-cancerous cell, so that you would not expect to see cancers forming. So as we've established, tumor suppressors are recessive at the cellular level, but something I haven't overtly stated is that if you look in families that are segregating a tumor suppressor, the pattern of inheritance looks like an autosomal dominant inheritance pattern. So for example, the BRCA1 pedigree that was studied by Mary Claire King, this is just one of the pedigrees that was looked at. When you look at the pedigrees in aggregate, it turns out that they exhibit an autosomal dominant inheritance pattern. So that's a little bit puzzling. But then if you look at the mutant alleles of the BRCA1 gene at the cellular level, they're in fact recessive at the cellular level, just like the examples I talked about in the preceding slides. Also, if we look at the mutant alleles of BRCA1 that segregate within these families, the mutations actually look like loss of function alleles. And typically loss of function alleles behave in a recessive fashion when you look in pedigrees. So this is a bit of a puzzle. I'll try and explain this puzzle over the next few slides. Another feature of tumor suppressors that I'm also going to try and explain is that they often display incomplete penetrance. So we'll also try and explain that over the next few slides. Okay, so why is it that tumor suppressor mutations exhibit a dominant inheritance pattern in pedigrees, but yet they exhibit a recessive pattern at the cellular level? It's very difficult to answer that question within a single slide. I'll just say what the simple answer is that unlike other recessive diseases, other diseases that exhibit a recessive inheritance pattern, such as, for example, cystic fibrosis, one thing that distinguishes cancer from a lot of recessive diseases is that cancer arises from a mutation that occurs in somatic tissues within a single cell. That's one thing that distinguishes it from things like cystic fibrosis. So, for example, if you've got a tumor suppressor segregating within a family that's showing it some sort of dominant inheritance pattern, typically what's segregating within that family is a heterozygous mutation in a tumor suppressor gene. So people start off life with carrying one mutant copy. In my figure here, the mutant copy is designated by the TS-, which is a tumor suppressor mutation. And they have one wild-type copy of the tumor suppressor gene. So as long as you have one functional copy of the tumor suppressor gene, you can actually maintain normal cell growth and proliferation under these conditions. But what can arise at some point during development in a somatic tissue is that a mutation can occur in the normal copy of the tumor suppressor gene such that it becomes inactivated at some point. So somatic mutations are just mutations that occur within the body tissues, not in the germline. So at some point during development, a mutation could occur that just inactivates the normal copy of the tumor suppressor. And when that happens, the cells that actually acquire that mutation and their descendants are no longer heterozygous. So that's called loss of heterozygosity. Now they actually have two defective copies of the tumor suppressor. And under these conditions, when you've got two defective copies, cell growth and proliferation are no longer under normal control, and the cells can start to grow and proliferate in an uncontrolled fashion. So now you're taking a step towards the cancerous state. There are a number of ways you can detect loss of heterozygosity. So let's, for example, consider the BRCA1 gene, again, a classic example of a tumor suppressor. And within the BRCA1 gene, there's actually a CAG repeat that's polymorphic in the population. So people in the population have different numbers of CAGs at this particular site. And so you could actually design a PCR test where you have primers that flank the CAG. You can perform a PCR experiment where you run out the alleles on an agarose gel. And then if you do that in a number of patients that have mutations in the BRCA1 gene or cancer caused by mutations in that gene, and you look in a number of different families, so for example, individual 1, 2, 3, and 4, as shown in this example here, if you look at the number of alleles of the CAG repeat in healthy tissues from, for example, individual number 1, and in the cancer tissue, what you'll see is in the healthy tissue, since the BRCA1 gene is an autosomal gene, in the healthy tissues you can actually detect two alleles. In the individual 1, for example, one allele has 300 base pairs, the other allele is 150 base pairs. And also, if you look at the very bottom of the figure, if you ask, can we detect BRCA1 protein in, say, for example, a biopsy tissue from the healthy tissue of this individual, the answer is yes, you can detect that protein. It's actually being made. However, if you look in the cancer tissue, what you notice in individual 1, you only can detect one allele that's at 300 base pairs. And if you ask whether the biopsy from the cancer tissue also expresses the BRCA1 protein, you can see that the answer is no. And then if you look in several other individuals, individual 2, and you do the same kind of analysis, individual 3, individual 4, what you might notice is that in individual 3, you also see only one of the two alleles in the cancer tissue. So in these two individuals, this is a classic example of what happens during loss of heterozygosity. You start off life with one defective copy, one normal copy, and at some point, the normal copy becomes inactivated in the cancerous tissue. So what's going on in individual 2 and 4? It may be that in individual 2 and 4, the mutation that inactivates the normal copy of the BRCA1 gene is just a small, it could be a coding mutation. It's probably a less severe allele, whereas in individuals 1 and 3, it could be that the mutation leads to a large deletion or some other kind of rearrangement. Okay, so in the individual 1 and 3, in the cancer tissue, you only see one allele. So now I've got a question for you. So the box allele in individual 1, is this allele associated with the wild type BRCA1 allele that was segregating in the family? Or is this allele associated with the mutant BRCA1 allele that was segregating this family? So in other words, is the box allele the normal allele that was inherited by this individual, or was it the mutant allele that was segregating within the family? So you can pause the video to think about this for a minute, and then I'll just restart it when you're ready to see the answer. So the answer is, this is the mutant BRCA1 allele that was segregating within the family. So the mutant allele, when you start off life, you inherit this mutant allele from one of your parents, and this mutant allele is inactivated right from the very start. And then what happens in the individual, in the cancerous tissue, is you actually lose, or you have some sort of mutation that inactivates the wild type BRCA1 allele that you inherited. So the one that's the polymorphisms that's missing corresponds to the wild type allele that you inherited from one of your parents, and it's been lost as a result of some sort of somatic mutation at some point during development. So these are some more examples of your ability to detect loss of heterozygosity. So to sort of walk you through this a little bit, so we've got two generations shown here. Individuals with black filled circles are people that have cancerous tissue, cancerous breast and cancerous ovarian tissue. Individuals with unfilled circles are individuals that have that are normal, they don't have cancer. And then within the individuals that have cancerous tissue, what I'm showing you is in the black filled circle is a biopsy and molecular analysis of tissue that's non-cancerous, as well as the other two circles represent biopsies from tissues that are either cancerous breast tissue or cancerous ovarian tissue. So if you do some sort of molecular analysis, if you look at the non-cancerous tissue in individual 2, for example, you can see that they have two different alleles of polymorphic markers that are spread along a certain length of the chromosome. But if you look at both the cancerous breast tissue and the cancerous ovarian tissue, what you can see is one of the two alleles along this length of chromosome that's being looked at, one of the two alleles is missing. So you've lost one of the two alleles along a certain length of the chromosome. And if you look at individual 1-3, you see kind of the same pattern emerging. And then if you look in generation 2, individual 1, you also see it's a slightly different pattern, but essentially this individual is missing two alleles of a particular marker along the length of the chromosome. So what this indicates is that at some point in development, some sort of mutation has arisen that's led to either some sort of large deletion or some other event that's taken place where you've lost either one portion of the chromosome or an entire chromosome, and you're now left with just defective copies of the tumor suppressor gene. There are many different mechanisms that can give rise to loss of heterozygosity. For example, you could have a non-disjunction event that takes place during mitosis, such that the normal functional copy of the tumor suppressor is lost because the chromosome that bears that is lost during a non-disjunction event. You could also have a deletion or insertion mutation in the functional copy of the tumor suppressor, such that now you have both copies of the tumor suppressor gene and both homologs are inactivated. A point mutation could arise in the functional copy of the tumor suppressor that inactivates it. You could also have mitotic recombination occurring as well. And then there are other things that could give rise to loss of heterozygosity. Now, I think you're pretty familiar with some of the first things that I've listed on here, non-disjunction, deletion, and insertion mutations, and point mutations, based on things that have been talked about earlier in this course. But one thing that we haven't talked about previously is mitotic recombination. So I'm just going to spend a few minutes walking you through what is involved in mitotic recombination and how this can lead to loss of heterozygosity. All right, so what's shown here is this is a cell that's just ready to undergo division. Basically, each of the homologs have replicated, so each of the different homologs are represented by different colors here. And the homolog with the mutation in the tumor suppressor gene is designated in sort of an orange color, and the homolog with the normal functional copy of the tumor suppressor is designated by green. So each of these lines represent a sister chromatid. And what can then happen before the cell divides is you could have a crossover event that takes place. So now I know you guys talked about crossover events a lot that happen during meiosis, and we spent a lot of time talking about the crossover events that lead to crossing over genes in the formation of gametes, and how you can use that to carry out linkage analysis and map genes. But what we haven't talked about previously is that this actually does rarely but occasionally occur during mitosis. And when this happens, let's say that this sort of crossover event that's pictured here has taken place, what happens is these sister chromatids will exchange portions, and then before cell division occurs, the sister chromatids look like this, where each of the sister chromatids is no longer identical. For example, the top homolog, you have one sister chromatid has a dysfunctional copy of the tumor suppressor, and the other sister chromatid has a functional copy of it. Likewise for the other homolog. So when spindle attachment takes place, okay, and by the way, this just to introduce some terms, I forgot this would pop up. This is mitotic recombination, and this actually happens oftentimes in mitosis to repair double-strand breaks. So double-strand breaks occur occasionally from things such as ionizing radiation, and mitotic recombination is a way that you can actually repair these breaks. We're not going to spend any time talking about that in this class because we just don't have enough time to talk about DNA repair. But anyway, when spindle attachment takes place, as the cells are getting ready to divide, And remember, this is mitosis, not meiosis. So again, if this is meiosis, the homologs would still be paired at this point, but because this is mitosis, the homologs are not paired with one another. And there's a couple of different ways that spindle attachment could occur. I'm just showing you one way that it could occur because this makes the point for loss of heterozygosity. So let's say spindles attached in this fashion. When cell division occurs, you're going to end up producing these two cell types, where one of the cells has two defective copies of the tumor suppressor gene, and the other cell division product has two functional copies of the tumor suppressor gene. So this cell that has two non-functional copies of the tumor suppressor can now go on to form ultimately a cancer cell. Let's try now to talk about resolving this paradox. As I mentioned already, the pedigrees of people that have tumor suppressors segregating in the family show a dominant inheritance pattern, but the mutant tumor suppressor is recessive to wild type at the cellular level. So this is a bit of a puzzle, and this is not something that's seen in other recessive disorders. So for example, cystic fibrosis at the cellular level is recessive, but it's also recessive in pedigrees as well. So the reason that cancers, the unique feature of cancers that gives rise to these characteristics is that unlike other genetic disorders, loss of heterozygosity in a single cell can ultimately lead to cancer. So cancer is just a series of mutations that occurs starting with a single cell that leads ultimately to a cell lineage that has a capacity for unregulated cell growth and cell division. With other recessive disorders, such as cystic fibrosis, for example, that's not the case. You could have heterozygous mutations of the cystic fibrosis gene that go on to undergo loss of heterozygosity. But if most of your cells in the lung are functioning okay, they have at least one functional copy of the cystic fibrosis gene, that doesn't have any effect on lungs function overall because there's hundreds of billions of cells in the lung. So why do mutations in BRCA1 and other tumor suppressor genes exhibit incomplete penetrance? So now we'll talk about that a little bit. Does it take only a single base change to cause cancer? Turns out the answer is no. People have studied the kinetics of transformation, and it turns out it looks like multiple mutations are required. In fact, perhaps as many as 10 mutations are required for a cell to eventually go on to become a cancerous cell. So let's imagine this is a cell lineage here, and each of these dots represents a cancer-promoting mutation. So typically what's required is you have to have a cell that undergoes that first mutation. So once you have that first mutation, the process is initiated, but you typically have to acquire additional mutations. So let's say in this example, you need to have 3 mutations for the cancer to develop. So in this case, it's basically not until you've acquired all 3 mutations in that same cell lineage until you actually develop the cancerous state. And as I said, it's thought that for many cancers, as many as 10 mutations are actually required. One of the things I mentioned early on at the beginning of the lecture is that cancer is a multi-step progressive loss of homeostasis. So basically what happens is that you have a initial mutation that occurs that might reduce growth control. So the cells are starting to divide a little bit more proficiently. And then you have additional mutation that occurs that leads to further loss of growth control. You might have some DNA replication errors that start to take place. There might be a failure of programmed cell death that takes place. Maybe telomerase activity is reactivated because telomerase activity, the activity that helps maintain the ends of DNA, it diminishes in somatic cells, and that might get reactivated at a certain point. And then eventually the cells acquire the ability for invasion and metastasis to travel to another site and start growing in another location in the body. And that leads ultimately to the formation of full-blown cancer. So it turns out each of these steps towards cancer is caused by additional somatic mutations. You need to start off with that initial mutation in one single cell, and then further mutations in the lineage that's derived from that initial cell leads ultimately to cancer, but it requires a number of mutations to get to the full-blown cancerous state. The inheritance of a tumor suppressor mutation confers a predisposition to cancer, but does not mean that you will definitely develop cancer. So what does predisposition mean? It's probably easiest to explain predisposition using our made-up example. So let's say it takes 3 specific mutations for a particular cancer to develop, as we talked about in this example. The mutation rate is about 1 mutation per 10 to the 7th bases polymerized per cell per generation. So that's a pretty low error rate. And given this low error rate, you have to have 3 improbable events have to occur in the same cell lineage. So for our made-up example here, the 3 mutations that occurred in this lineage that's highlighted in yellow. So it's not just 3 mutations that have to occur in that lineage, but 3 mutations in critical genes that would give rise to ultimately a cancerous cell. So these are 3 very improbable events given that the mutation rate is very low and that the only mutations in certain genes would give rise to cancer. So in families that actually are segregating a tumor suppressor mutation, it turns out they're already starting off life with 1 mutation that's inherited. So basically, this is a situation where instead of 3 mutations having to take place, only 2 mutations now have to take place in these individuals. So the set of events becomes much less improbable if you've inherited one of the mutations right from the start in life. But the thing is even with individuals that have inherited a tumor suppressor mutation, it's not a guarantee that you're still going to get the remaining set of mutations that have to occur to cause this cell to drive all the way to the formation of a cancer cell. And that's why tumor suppressors aren't fully penetrant. It just really depends on whether these other mutations can occur in the cell lineage in genes that would be responsible for driving the cell towards the cancerous state. Okay, to summarize the key points that I've been talking about up till now, cancer is caused by mutations in genes that typically regulate the cell cycle. Mutations in primarily 2 gene classes predispose to cancer. Proto-oncogenes, these are cell cycle activators, and mutations often increase gene activity. They're dominant, gain of function at the cellular level. And tumor suppressors, these are cell cycle negative regulators. The mutations decrease gene activity. They're loss of function mutations. They're recessive at the cellular level, but dominant in pedigrees. And finally, multiple mutations within the same cell are required for cancer development. It isn't just 1 mutation that occurs and you've got cancer. It requires multiple mutations, as many as 10 perhaps. So we've now spent a lot of time talking about the sorts of mutations that give rise to cancer. So I thought it would be worth it to spend a little bit of time talking about how knowledge of a cancer-causing gene defect might impact treatment for cancer. So I'm going to tell you 3 stories. The first story is about Angelina Jolie, at the time Angelina Jolie Pitt. And this is a story that was published in the New York Times around 2015. Basically, the story goes as follows. Angelina Jolie had several relatives that had died of cancer, including her mother, her grandmother, and her aunt. And she had a blood test done, and they found in the blood test that she actually had a mutation in the BRCA1 gene. And it turns out that the particular mutant allele that she had gave her about an 87% risk of developing breast cancer and about a 50% risk of ovarian cancer. So she definitely faced a very difficult challenge here and a very difficult choice. But she ended up deciding to have a double mastectomy to greatly reduce her risk of developing cancer. So I'm sure it was a very difficult decision she had to make to kind of decide whether to do that. But the fact that she actually knew her cancer risk gave her the opportunity to at least do something about it and to do something that would reduce her risk. All right, the second story is a little bit more complicated now. This is a case of a woman, Angie Watts, who had been diagnosed with breast cancer, and she was in the process of trying to decide how to treat the breast cancer. They did a blood test on her and found that she also had a mutation in the BRCA1 gene. One of the things I just mentioned very quickly, it turns out BRCA1, again, acts at a checkpoint. And checkpoints, genes or regulators such as BRCA1 oftentimes also help make the decision about whether, if the DNA is heavily damaged, whether the cell should undergo a form of programmed cell death called apoptosis. So because she had a mutant allele of BRCA1 gene, the concern was one of the reasons that people do use radiation therapy to treat cancers is because you're trying to basically cause DNA breaks that would trigger the cancer cells to undergo apoptosis. But she had a mutation in the BRCA1 gene. The concern was that the cells may not be able to undergo apoptosis. And the downside of treating cancer cells with radiation therapy is it creates new mutations. And so it could actually end up not doing what they would hope, which is the cells would undergo apoptosis, but instead it would create new mutations that might make the cancer more aggressive. The problem is that the allele of BRCA1 that she had wasn't really very well studied, so they didn't really know what the effects of that mutant allele were, whether it would inactivate the gene or whether it really had no effect on the function of the gene. So the doctors couldn't really agree whether it was going to be a benign allele or whether it was going to be a loss of function allele. So they kind of left it up to her to decide what sort of therapy she should pursue. So that's kind of a difficult decision when you don't really have good information to go on. And this third story is probably the happiest story. This is a story about Lucas Wortman. He actually was a doctor who was studying leukemia, and he actually ended up developing leukemia himself. And one of the things he decided to do once he discovered he had leukemia is he tried to get some friends who were doing next generation sequencing to sequence his cancer genome to kind of figure out what gene defects he had, hopefully in an effort to kind of come up with a more tailored therapy that would be able to treat the disease based on the mutations he was carrying. So what Dr. Wortman and his colleagues did is they took a leukemia cell from Dr. Wortman and a normal skin cell, and then they collected DNA from both of these cells, and they sequenced the DNA in those cells and compared the sequence to look for mutations. Specifically, what they did is they performed exome sequencing, so they were only sequencing the expressed portion of the genome, the part that codes for protein. And then when they did this comparison, they did not detect any mutations in any genes that were known to cause cancer, so it did not produce any useful insight. So what they decided to do then was they decided to collect and sequence the RNA after converting the RNA to DNA. So I'm not going to tell you how you convert RNA to DNA. If you've taken a biochemistry course, they might have covered that in that course. But there's no point in going through that here. I just don't want to belabor the point. Essentially, there's ways of converting RNA to DNA and then sequencing the DNA. So what they did is they collected RNA from the leukemia cell and from the normal skin cell. They converted it to DNA. Again, I'm just going to sort of present that like it's magic. And then they sequenced the DNA that correspond to the RNA, and they compared the sequences in the leukemia cell and the normal skin cell. And I should say, when you sequence RNA, this sequencing of RNA, you're not actually sequencing RNA, you're sequencing DNA that's made from the RNA. This is called RNA-seq. And before describing the results, I just want to talk a little bit about RNA-seq, just so to help you interpret the results. So this is a slide I presented from lecture 12. And what I told you about this slide, this is based on DNA sequencing, that sequencing read depth is proportional to DNA copy number. And I mentioned to you when I showed you this slide, is that by doing this kind of sequencing and comparing sequences between different individuals, it can help you to identify places where the DNA copy number is either reduced or increased. So for example, the sequencing read depth chart shown at the very top shows you a deletion. There's a portion where the sequencing read depth drops off at one portion of chromosome 11. The one in the middle is from a normal individual, and then the sequencing read depth chart at the very bottom shows a place on a particular individual where the sequencing read depth is higher. So this indicates there's a duplication of a portion of the chromosome. So it turns out sequencing read depth is also proportional to RNA copy number. So you can use that to measure the total transcript abundance for any gene that you can detect based on the RNA sequencing that you've done. So to illustrate that, we can go on to the next slide. Let's consider 4 different genes represented by these 4 different colored boxes. As you can see, basically, each of these genes are producing messenger RNAs, designated by the straight lines below the arrows. And the number of messenger RNAs corresponding to each gene differs depending on the gene. For example, the blue gene is only producing 1 messenger RNA, the red gene is producing a number of messenger RNAs, and the other 2 genes are producing somewhere in between. So the way you would use RNA-seq to measure our mRNA abundance of each of these genes is as follows. Essentially, you would collect RNA from cells that are expressing these genes. You'd make DNA copies of the RNA. Again, I'm not going to describe how you do that in any detail. Then you'd fragment the DNA DNA that you synthesize from the RNA, and then you'd add sequencing adapters for next generation sequencing, and then you perform next generation sequencing on these samples. And then you'd map all these reads back to the genome. So you try and look at with the sequences and see where they correspond to the sequences in the genome, which genes they correspond to essentially. So if you did that kind of analysis, you might get results that look sort of like this, where, for example, the blue gene, each of these little blocks represents a sequencing read, and based on the sequence, the mRNA abundance of the genes, based on the figure at the very top, what you'd expect is you'd see a corresponding number of sequencing reads from these genes depending on how highly expressed they are. So sequencing read depth is proportional to transcript abundance, is another way of putting that. So for example, if we arbitrarily call the sequencing read depth of the blue gene 1, what we could say is that the green gene is producing twice as many sequencing reads, so it's probably expressed twice as highly as the blue gene. The yellow gene is producing 4 times as many sequence reads as the blue gene, and the red gene is producing 8 times as many sequence reads. So the number of sequence reads is basically proportional to the transcript abundance for each of these genes as well. So by looking at how many sequence reads you detect for each of these genes, it can tell you how much transcript abundance is between these different genes. So the results of RNA sequencing showed that mRNAs from a particular gene called FLT3 were overabundant in the leukemia cells from Dr. Wortman. So the results look sort of like this, where I'm just showing you 3 genes, where the red gene is the FLT3 gene. So in the normal skin cells, they found that the blue and the yellow genes were similarly expressed in the normal skin cells relative to the leukemia cells, but FLT3 was much more abundantly transcribed, or the number of reads was much greater for FLT3 in the leukemia cells relative to the normal skin cells. So it looked like the FLT3 gene was highly overexpressed in the leukemia cells. So the hypothesis that emerged from this is that FLT3 might be the gene that's somehow involved in the cancer in Dr. Wortman's leukemia. And there was already something known about FLT3, quite a bit known about it actually. So FLT3 encodes a cell surface receptor that promotes the formation of new white blood cells. So the FLT3 is on the surface of the cell, it binds to growth factors, and when growth factors bind to FLT3, it triggers the cell to actually grow and to divide. So the idea is that basically the FLT3 gene was overproduced in the leukemia cells, and because it was making too many receptors, that it was binding too much of the growth factor, and the cells were basically dividing at too high a rate. So this is what was driving the leukemia in Dr. Wortman's tissues. So fortunately for Dr. Wortman, it turns out that there was already a drug that would antagonize or inhibit the FLT3 receptor called sunitinib. And basically, it turns out that overproduction of FLT3 was already previously known to cause certain types of kidney cancer. It wasn't previously recognized as a cause of leukemia, but the studies of Dr. Wortman's leukemia cells indicated that it was probably causing this leukemia as well. So if you treat individuals with sunitinib, it basically blocks the FLT3 receptor so that even though you're overproducing the receptor, it can't bind the growth factor very effectively, and so the cells are not undergoing uncontrolled cell division, basically. So 2 weeks after taking the drug, tests revealed that Dr. Wortman's leukemia was in remission. So it looks like this was actually a very effective therapy for the type of cancer he had, and nobody would have even thought to try this therapy if they hadn't conducted this RNA-seq experiment to show that it was actually FLT3 that was overproduced and what was probably primarily driving the cancer in his leukemia. So the final question, very last slide. How would you classify the wild-type FLT3 gene? Would it be an oncogene, a proto-oncogene, a tumor suppressor gene, or both A and B? So you can stop the slide if you want, or stop the presentation and answer this question, but I'll just tell you the answer right now. So the wild type would be a proto-oncogene, and the mutant version would be an oncogene.